Good morning to all. Yesterday there was a disconnection from net and I couldn't join. So I think I was around this slide. So I will repeat this concept of association mapping. So there are two types of association mapping. One is candidate gene association mapping, where we have some prerequisite information about the candidate region under consideration. Candidate means probable region which may be playing role in that particular gene, particular phenotype or quantitative trait. So we limit our search to that region and we try to associate the polymorphisms present within that region for association with the trait. So there is one limitation of this approach that because we are limiting our search to particular region, there are other loci governing that particular trait. They are not accounted in this approach. But this is also a good approach when you have some prerequisite information about that particular region. The next method is genome-wide association mapping, where we search across the genome for the polymorphisms. And we perform LD mapping and we try to associate those LD maps with the phenotype. That's why genome-wide association mapping. In this approach, we try to consider as many as possible markers and we associate LD map data of these markers with the quantitative trait. Initially, this was not possible, but nowadays, due to advancement of this NGS and genotyping by sequencing technology, it is possible to search for the genome-wide polymorphisms, and now it is possible to create LD maps, which is genome-wide, where we consider more numbers of SNP markers, and we associate that data with the trait. This is also, this approach is possible with many traits also. Multiple trait analysis is also possible with the same set of markers, polymorphisms identified in particular population. A population might be germplasm panel, it may be other populations like nested association mapping population and MAGIC population, that is multiple advanced generation intercross. So these are the two populations utilized for combined linkage and LD mapping. As we know that linkage has advantage of detecting, it's good for initial detection, but it is poor for fine mapping, and association mapping or LD mapping is better for fine mapping, but it is not good for initial detection. So both the benefits of this linkage mapping and LD mapping, they are combined utilizing these two populations, NAME and MAGIC. So let us talk about NAME population. So I will directly go to the slide that was proposed in 2008 in maize. So in nested association mapping population, they utilize total 26 genotypes of maize. These are the 25 founder lines. And those founder lines were crossed with the common parent. The common parent is B73, and these founder lines were chosen because of their diversity and some of the important traits they are carrying related to seed quality, biotic, abiotic stress resistance, and so many things. So when each founder line, for simplicity, only one homologous pair of each founder line is shown here. So when we produce F1 of any two parent, it will receive one homologous chromosome from parent 1, another, the gray color chromosome, from common parent. So all these F1s will have common chromosome from common parent, the gray one. Other homologous chromosomes are varying because of the founder lines used for their development. Now each F1 population is handled according to single seed descent method, and 200 RILs were produced from each F1. So there are 25 F1s, so total 5,000 RILs were produced in this population. And because we have utilized multiple parent, there are multiple alleles present for a particular locus in these populations. So if you look at the single homologous chromosome pair, because of the recombination occurring in F1 meiosis and onwards, when the RILs are developed after, let us say, F6 or F8, up to F6 or F8 generation, this would be the situation of this homologous pair in each RIL. These are the numbers of RIL. And same thing is happening to other chromosomes also. This is for simple understanding, only one homologous chromosome pair is mentioned here. So you can see here, because of the recombination between this founder line and the common parent, we see blocks here, blocks of gray and maroon color, I think. So there are homozygous blocks present according to the concept of RIL development. These gray color regions and these blocks, they indicate linkage blocks. That means whatever real-time recombination occurred here, these blocks are present due to that real-time recombination. And you can see also recombination points or linkage blocks are different from one RIL to another RIL for the same homologous chromosome pair, up to 200 RILs. Same thing is happening to other chromosomes also. Similarly, here if you consider this particular cross, there are linkage blocks for this purple and gray color, and that represents real-time recombination. So utilizing that, we can perform linkage mapping, which is very good for initial detection. And within that limited region, which is not taking part in recombination, real-time recombination here, within this particular region, horizontally across these RILs, LD mapping can be performed, considering evolutionary and historical recombination, because within that region, recombination has not taken place during the development of RILs. So that way, this population can be utilized for combined linkage and LD mapping. When we use LD mapping, it gives good resolution. So here we are combining the initial detection power of linkage mapping and fine mapping power of association mapping. So that's called combined LD and linkage mapping, and for that this nested association mapping population is used. Why it is called nested? Because we are performing association mapping, it is called association mapping population. Nested because here if you look at this linkage blocks, then if you look at these linkage blocks, then within the particular block, if you look at this particular block from the founder line, then it is actually nested between these gray markers. And the markers which are present here, the polymorphism present here in these grayish boxes, they are common parent specific marker. So all the founder line blocks which are situated between this common parent specific marker, they are called nested between common parent specific markers present around this founder line region, which is shown by the color. So that's why it is called nested association mapping population. And here the genomes of founder lines are sequenced by utilizing next generation sequencing method, and the data of these founder lines are also deposited online. Data of the common parent is also deposited online, and data of these RILs are also deposited. So according to the trait you want to consider, suppose you want to perform association mapping in maize, they are ready to give these RILs. And you can obtain selected RILs also, not necessary that we study all these 5,000. Depending upon the trait under consideration, you can select the founder line and you can obtain RILs for those founder lines only. You phenotypically evaluate that population, and because these are the RILs, they are immortal population. Once they are genotyped, or let us say all the genomes have been sequenced utilizing NGS method, that genotype, genetic constitution is not going to change. Or otherwise, you can also follow GBS approach to genotype these RILs for SNPs, genome-wide SNPs. And then once you have the polymorphisms, once you have the phenotypic data from this RIL under particular situation, you can perform association mapping, combining the LD map data, linkage map data, and the phenotype. So that's how nested association mapping is utilized to perform linkage mapping and LD mapping simultaneously, or association mapping simultaneously. Now, the next population utilized for combined linkage and association mapping is MAGIC, multiple advanced generation intercross. So in Bendilo et al. At Philippines, ERI, 2012, they first time developed this kind of population in rice. So these are the genotypes they have selected, including this very famous mega variety Samba Masuri with sub-1 tolerance gene that is responsible for flood tolerance. This is a mega variety. The other parents they have selected because of the blast resistance genes they carry, high yield, drought tolerance, salt tolerance, and all these things. They cross these 8 parents in half dialyl fashion. So there are 8 parents. So there will be 28 processes developed if we follow half dialing design, considering the formula n into n minus 1 by. So here magic population is developed by crossing these 8 genotypes in half dialyl fashion. So we'll get 28 F1s. These 28 F1s are again crossed with each other. This would be the 4-way cross. And then finally 74-way crosses develop in such a manner they were also intercross. So 8-way crosses were developed ultimately. And from those 8-way crosses, from 35 combinations, total 60 plants were selected. The crosses were selected in such a manner that that cross carries all the genes from this 8 parent. So if you look at the single cross, you can see here it would be definitely heterozygous and it carries various segments from this parent. And then all these crosses were handled according to single seed descent method. And immortalized RILs were developed from these crosses. So if you look at the RILs developed, you can see the blocks. These blocks are narrow, so they represent the recombination due to this hybridization, which increases recombination. So these are the all linkage blocks present here, and within that block, alleles are not disturbed, and within that block we can perform LD mapping because the historical data are not disturbed due to real-time recombination. So this can be utilized for linkage mapping when we see these blocks, and within that block we can perform association mapping utilizing all the RILs developed by this kind of the crossing. That's why they are called multiple advanced generation intercross. Multiple because we are utilizing multiple parents. Advanced generation intercross because we are carrying out intermating in this 4-way and 8-way manner. And then finally RILs are developed. So this population is known as magic population. This intercrossing is intercrossing is carried out to increase the recombination rate so that more and more fine mapping is possible. So these two population, NAME and magic, are utilized for combined LD and linkage mapping. And same group also validated that magic population by carrying out the association mapping for known locus, that was sub-1 locus present in Samba Masuri. So they utilized that parent. So in that population, sub-1 locus was present. They evaluated all these magic RILs for submerged tolerance, a particular phenotypic screening method, and they genotype these RILs with SNPs. So these are the colors representing the chromosome from 1 to 12, because we are talking about rice. These are the log P values, as I mentioned yesterday. This is a Manhattan plot for association mapping. And this is the mixed linear model taking into consideration population structure and genetic relationship. This is the threshold value for the error probability, which is 4. As this value increases, the error probability decreases. So we are getting more and more significant SNPs associated with the trait if the points goes above threshold value. So here you can see the sub-1 locus, it was already known that it is present on 9th chromosome, and they found the highest peak of this SNPs having the p-value of 9.5, around 9 to 10 for that particular locus. So that's how they validated this population, that this also shows the significance for already known locus. So these kind of the populations can be utilized for association mapping. So now again the same table I will discuss. I have already gone through this biparental population, the differences between them. When we compare magic and NAM populations with this, if you compare the parents involved, there are multiple parents involved. Resource type is immortal because in both this population we develop RILs which are highly homozygous. Mapping individuals, they are homozygous, magic and NAM population. Generation requires, because in this we perform crossing and then we follow RIL development program. So it is about 8 generations required. Here for, sorry, in magic, more generations are required because intercrossing is carried out, and in name population, 6 to 8 generations are required. Here we require more than 8 because of the intercrossing carried out in half diallel manner. And once the 4 ways and 8 ways processes are developed, after that we start developing RIL. So it requires more than 8 years. Multiple trait analysis and multiple analysis is possible because now we have multiple parents. So there is a possibility of having multiple alleles here. Recombination events are very high in both this population. It is suitable for combined linkage and LD mapping. Mapping resolution is also very high because it takes into account linkage as well as LD. Replicated studies are possible because our material is homozygous and immortal. Minor QTLs can also be detected in this population. So these are the advantages of MAGIC and NAM over biparental population. So that's all about linkage mapping and association mapping. Now I will wait for 2 to 3 minutes if there is any question related to yesterday's lecture or today, and then I will proceed with marker assisted back crossing. Okay, then if there are no questions, then I will continue with marker assisted back crossing, and then we'll take into considerations if there are any questions. So once we perform gene mapping, we identify the markers which are either linked with the gene of interest or QTL, or we identify the markers, the polymorphisms which are associated with particular quantitative trait. Once we do that, we have the markers which are linked or associated with the trait. Utilizing those markers, we utilize those markers in various breeding programs. So the first method, most widely utilized method for transferring qualitative trait by utilizing markers is marker assisted back crossing. So I have just kept a few terminologies here which are very preliminary for breeding. That means when we say back crossing, we are crossing utilizing emasculation and crossing, pollination. And back cross means we cross F1 with either of the parent, and we apply selection So selection means differential reproductivity and viability. It's natural and artificial both. And when we use marker for this kind of the selection, it becomes marker assisted selection. So in marker assisted selection, we utilize linked polymorphism for indirect selection of a particular trait. So that linked polymorphism is DNA marker, and the process is called marker assisted selection. We can apply this marker assisted selection in any breeding method. F2, backcross, recurrent selection, gene pyramiding, there are many. We'll go through them one after other. So this is conventional backcross, where we have one high-yielding elite cultivar, which is susceptible to particular disease, let us say, and we have one donor parent, which is resistant to particular character. Here we are talking about flood tolerance. So this is high-yielding variety, but susceptible to flood. If this is submerged under the flood, under the water, then it will give very poor yield. But we have a donor which is submerged tolerance or flood tolerance. So we cross them. These are our P1 and P2. This is recurrent parent. This is non-recurrent parent. This is recipient parent. This is donor parent. So once we develop the F1 in conventional backcross method, we'll cross this F1 to recurrent parent, where we want to transfer the trait from donor. So by recurrent backcrossing, what actually we are doing, we are transferring most of the genes from this P1 recurrent parent to F1 nucleus. That's what we are doing. But we are retaining one trait from the donor, target trait, that is flood tolerance here. So in each backcross progeny, we'll perform screening, and only flood tolerant progenies will be crossed again to the parent. And after backcross 6, we perform selfing. That would be backcross 6F2. And we try to select the individual here, which is having maximum genetic contribution, 99.9% from recurrent parent, and one target locus, which is from donor parent. And if we are transferring some of the traits, there might be problem of linkage drag, where undesirable traits are linked with our desirable trait from the donor parent. When there is a problem of linkage drag, we have to increase the number of backcrosses. It may go up to 20 to 25 backcrosses, depending upon the level of linkage, type of linkage, whether it is loose linkage or tight linkage. Accordingly, we have to increase the number of backcrosses. So these are the advantages of marker-assisted selection in general before going to marker-assisted backcrossing. Whenever conventional breeding for a particular trait where the phenotypic screening is very difficult, like in flood tolerance and abiotic stress, salinity stress, growth tolerance, they are very expensive. Phenotypic methods are very expensive and time consuming also, tedious also. So in that case, we can go for marker-assisted selection, where we do not apply phenotypic screening, but once the marker is identified, we utilize this title link marker for indirect selection of the gene of interest. Sometimes some of the traits are only observable at maturity. So for phenotypic selection of these traits, you have to wait for the maturity, and you cannot utilize the backcross, because when the progeny is mature, they would not have flower in most of the crops, especially determinate crops like maize, rice, wheat. So if these are the seed characteristics, then you will have to wait for the maturity, and that will create problem in crossing programs. So if you have the markers which are associated or linked with a particular trait of interest, then the seedling stage itself, we can identify the individuals carrying the desirable gene from donor parent, and we backcross those progenies to the parent. More the complex trait, in case of conventional breeding, more time, efforts, and cost are involved for phenotypic screening. So for complex trait, like abiotic stress resistance and other, marker-assisted selection may be helpful. So the major goal of marker-assisted selection is to reduce time and cost, and also the individuals you are handling. It also reduces the population size to be handled. And because codominant markers are available, we can distinguish heterozygous individuals from both the homozygotes. So I will just give you one simple example of benefit of marker-assisted selection. In conventional backcross method, we have two methods: marker-assisted backcrossing to transfer dominant allele, and backcrossing program for transfer of the recessive allele. So in one case, we transfer dominant allele through conventional means, and in another case, we have recessive allele to be transferred. So backcrossing program for transfer of dominant gene is very simple. Every after every backcrossing, you just backcross progenies, selected backcross progenies having the target trait to the recurrent parent. But when you transfer recessive gene to the recurrent parent, then in that case you have to go for selfing after every first, third, fifth backcross program, because the recessive allele is only expressed phenotypically under homozygous condition. So when recessive allele is not present in the homozygous condition, in particular backcross progeny, you cannot apply selection. So we have to carry out selfing, and that increases number of backcrosses to be done. If you have marker which is linked with the recessive allele, then this selfing is not required. In the backcross 1 itself, you can identify the heterozygous plant, and you backcross that heterozygous plant with the recurrent parent, because you know by marker selection that these plants are carrying the recessive allele. In conventional backcross program, you can only know when you carry out selfing, when you go through backcross 1 F2, then and then you can do this. So if you utilize marker, backcross program for transfer of dominant gene, backcross program for transfer of recessive gene are similar. They take the same time. So that's the advantage. This is the one thing about marker selection, one benefit. So this is the conventional plant breeding. If you are applying selection in F2, you phenotypically screen the plant and you select the individuals. So this is what we do in conventional breeding program, where selection is based on phenotype, and phenotype is outcome of genes and environment both. So environment will also incorporate errors in this selection. And for some of the complex traits, the selection will be very difficult and costly and tedious. But for marker-assisted selection, we take the leaf samples from the progeny in the seedling stage itself, and then we extract the DNA, and then we identify the polymorphism, marker polymorphism, which may be PCR and electrophoresis based, or which may be sequencing based. And then we analyze the markers for their presence and absence, and we genotype the plant, and then we select the plants accordingly. So in marker-assisted selection, we are focusing on the markers. This is the electrophoresis pattern. Rather than selecting the individuals phenotypically, we select individuals on the basis of marker. So only few individuals are carried forward according to the marker phenotype. Marker genotype. And ultimately, the number of plants you will be handling in the next generation are very small in numbers, so that is easier for a plant breeder. Advantages of marker selection: it is easier wherever phenotypic screening is difficult, because in marker selection generally we do not apply phenotypic screening when we have a tightly linked proven marker. So we are free from the laborious phenotypic screening method, which saves time and resources both. Selection at seedling stage is also possible, because marker assisted selection is not dependent upon particular stage of the crop growth, because we are directly dealing with DNA. So if a seedling is carrying that resistant allele or gene, definitely it will show resistance to particular disease at any point of time in that plant cycle, life cycle of that plant. So if you are, as I told you earlier, if you are selecting for grain quality, you can also apply selection at seedling stage. You need not to wait for flowering and maturity. And you can apply also selection before transplanting of rice, if you are talking about rice, for the grain quality or any other quality. And of course, grain quality is very important in case of rice. Because we are utilizing DNA markers, here there is no environmental influence. So marker selection is more reliable as compared to conventional phenotypic selection applied in plant breeding. And it can distinguish heterozygous from both the homozygous. So there is a reliability. If we compare a simple pedigree method where only phenotypic selection is applied and coupled with marker assisted selection, you can see here in a conventional pedigree method, we will be selecting many plants from F2, F3, F4 generations. But in marker assisted pedigree method, you'll be selecting only few plants carrying the desirable genes. So the material to be handled is reduced. You can compare the number of lines and progenies you have to maintain. So that, see, the time duration is same when we apply marker selection in pedigree method in this particular case. But the material to be handled is very small. So that increases the efficiency of selection and material to be handled. But nowadays, the time duration can also be reduced by utilizing SNP markers and genotyping by sequencing. So as I mentioned earlier during QTL analysis, the same concept also is applicable for marker-assisted backcrossing. We utilize this concept in one of the component of marker-assisted backcrossing. So I'm explaining it again. So rather than one marker, two markers, flanking markers are always better. Because if we utilize one marker, as I explained earlier, suppose this marker is 5 centimorgan away from this particular gene, we are transferring through backcrossing here in this particular PPT, this particular lecture. So efficiency of marker is 95%. It is simply 1 minus the recombination between marker and the gene of interest in decimal terms. So it is 95%. It means the 100 backcross progenies you will be selecting on the basis of marker, in 95 progenies, marker will be definitely coupled with our target gene. In other remaining 5, there is no surety. That's the meaning of 95% confidence and 5% level of significance. If we utilize two flanking markers to our gene of interest, which are 5 centimorgan away from the gene, situated on both the sides, that is the meaning of flanking marker. Here we can work out the efficiency by utilizing this formula, 1 minus 2, recombination between a marker A and gene, marker B and gene of interest. This is our gene of interest. So if you work out, it is 99.5%. That means out of 200 backcross progenies you are selecting, 199 progenies, 200 backcross progenies you have selected on the basis of presence of marker A and B. So out of 200 progenies you have selected by utilizing these two markers, 199 progenies will definitely have the traits. So that kind of increment can be achieved by utilizing two markers rather than single marker. Definitely when you utilize two markers, your cost will increase, but that is giving you the reliability. So you have to weigh the accuracy and cost. So marker-assisted selection is especially useful when the trait is governed by recessive gene. When there are multiple genes governing the disease resistance, as it is the case in case of wheat rust, where there are multiple disease resistance genes, again many RES-specific genes are there. If you want to pyramid all these genes in one genotype, then also marker-assisted selection can be useful when the trait is governed by quantitative trait and when there is large gene-to-E interaction. In all these cases, marker-assisted selection will be more useful, reliable, and less time consuming as compared to conventional selection. So there are many schemes for marker assisted breeding. Early generation selection, as I explained, the pedigree method. Marker assisted backcrossing, we are going to discuss today. Marker assisted gene pyramiding, marker assisted recurrent selection, genomic selection, and combined approaches. So let us consider marker assisted backcross. So over conventional backcrosses, marker-assisted backcrossing has certain advantages. And there is an effective selection of the target locus. By utilizing a tightly linked marker to the target locus, we can effectively select the target locus because we are directly dealing with the DNA, actual cause of the variation of the phenotype, which may be resistance to disease or abiotic stress or any. So there is effective selection for the target locus, and that component is called foreground selection. When you select target gene in the backcross progenies by utilizing a tightly linked marker to the target locus. It is called foreground selection. It minimizes the linkage drag by utilizing two flanking markers surrounding the target locus. I will talk about this. It is called recombinant selection. Especially this is utilized to minimize the linkage drag. And the background selection, where we select the background of the backcross progenies. Maximum number of the loci should be from recurrent parent. That's the meaning of background. So background selection is utilized to accelerate the recovery from the recurrent parent. More and more alleles, if you genotype the backcross progeny carrying the target locus, if you genotype this backcross progenies for this background markers, generally these are the microsatellite markers. If most of the alleles are from recurrent parent, then you are sure that the recovery of recurrent parent is higher. So sometimes both these two components are categorized under background selection, but I would distinguish foreground selection, recombinant selection, and there is a distinction between recombinant selection and background selection also. I will explain them. So these are the three important aspects of marker-assisted backcross: effective selection of target locus, minimize the linkage drag, and to accelerate the recovery of recurrent parent. Generally, this to get the 99.9% genome of the recurrent parent, 5 to 6 backcrosses are required. But by utilizing marker-assisted backcrossing, we can achieve that within backcross 2 or backcross 3. These are the essential requirements of marker-assisted backcrossing: a tightly linked marker to the target locus for foreground selection, two flanking markers, generally 5 centimorgan away from the target locus for recombinant selection, and background markers, markers which are present in all other chromosomes apart from the target locus, so that we can accelerate the recovery of recurrent parent. Let us consider the first component, which is foreground selection. In foreground selection, as I mentioned, we utilize the tightly linked marker with the target locus. This target locus we are transferring from donor parent to recurrent parent. So it is also applicable to dominant and recessive gene equally. Once we have the link marker, tightly linked marker, we can utilize that for selection of the target locus. So that's foreground selection. So we apply foreground selection in each backcross generation, backcross 1, 2, 3, and onwards. Now the linkage drag. So linkage drag means undesirable genes which are tightly linked with the target locus. These are the undesirable genes. Even if you reach up to backcross 10 in conventional backcrossing program, these genes cannot be removed because they are tightly linked. More tighter the linkage, more and more backcrosses are required to eliminate this undesirable linkage. So if that linkage is very tight, it means the undesirable genes are tightly linked with the gene of interest. It may require 20 backcross generations to eliminate this linkage drag. But if we apply marker-assisted backcrossing and recombinant selection, that can be done within backcross 2. So that's the power of marker-assisted backcrossing. You are saving time, cost, and you are coming with the variety very quickly. I will explain how this is possible with marker-assisted backcrossing. So this is how we minimize the linkage drag by recombinant selection. We need two flanking markers shown by these blue arrows. This is our target locus, and we need two flanking markers which are 5 centimorgan away from the target locus. So closer these markers to the target gene, we need to open the large population in backgrounds. Now, if we look at this flanking polymorphism between parent 1 and parent 2, the double crossover between these two regions here, one crossover here and one here. This double crossover will transfer this particular target locus to the chromosome of recurrent parent. It may happen within backcross 1 or backcross 2. But if you are expecting double crossover here, or this recombination to occur, it is very less likely because the frequency of double crossover is very low. And if you are expecting that in Becros 1 itself, it is possible. But in that case, you have to open a very large number of plants in Becros 1 because these are linked with the target locus. These flanking markers are linked. So in that case, you may have to open up the population of 5,000 or 10,000 individuals, depending upon the distance between this flanking marker and the gene. So the genotyping this 5,000 backcross progeny, generating this 5,000 backcross progeny and 10,000 progenies is tedious, and it is costly also. So what is done to reduce the population size? We perform sequential selection. First backcross 1, we try to look for the recombination in this region, and in backcross 2, keeping this constant, we try to select the recombination in another region. So let us see how it is performed. This is our target locus, and these are the two flanking markers. Let us say 5 centimorgan away. There is a marker polymorphism between parent 1 and 2, recurrent parent and donor parent for these two loci. So in backcross 1, you may apply selection for any of this marker polymorphism. Let us say in one case we focus here. So in backcross 1, we select the individuals where recombination has occurred between target locus and this marker. So if we have applied selection for this in backcross 1, then the backcross 2 we will keep this constant. That means target gene must have the allele from recurrent parent. That indicates that the recombination has occurred between the target region from the donor and the segment from or the chromosome of recurrent parent. So in backcross 2, we'll apply selection for recombination here. I will also explain the next coming slide how we do this considering the markers. And in backcross 1, if you are focused on this side first, then in backcross 2, you will focus on another side, keeping this constant. So what it is indicating that recombination has occurred between chromosome of recurrent parent and this target gene in the region closely situated around this target locus. So that's how linkage drag is minimized. Or we can select the progenies where this recombination has occurred. Now, how marker profile can give you idea about the recombination? Because here we are not applying phenotypic selection. So to understand that, I have kept this slide. Let us say these are the two alleles. This is associated with resistance, this is associated with susceptibility. And we develop F1. These are the markers linked with the resistant locus. So this is a codominant marker, and F1, both these alleles are present. And if you consider the F2 individuals from this, then you can see this allele is present similar to parent 1. So this marker allele present indicating the resistance. When both the alleles are present, individual is also resistant. So it can be concluded here that the resistance is governed by dominant allele. So heterozygous individuals are also showing the resistance. And whenever this particular allele is present, here you can see only this allele, it indicates susceptibility. But there are few cases where only this allele is present, you can see here. But when you perform phenotypic screening, actually the plant is resistant. So how that has happened? That has happened because of the recombination between this marker locus and the resistant locus in such a way that marker allele linked with the susceptibility has been transferred. Because of the recombination, it has been transferred to the resistant allele. So now resistant allele is present with this particular marker allele due to recombination between marker locus and gene of interest. So these are the recombinants, and that's how they can be identified in any segregating generation. Now let us take the example of XA21, marker assisted backcrossing for XA21, which provides resistance to bacterial blight. So this is our recurrent parent. This is our donor parent, IRBB24, which has this dominant gene, which provides resistance to Xanthomonas. And there are two alleles in homozygous condition, indicated by this red color. This is also homozygous. This is the target locus. These are the flanking markers. The capital L and capital B are alleles of two marker loci flanking this target locus. This is donor parent. M63 is our recurrent parent carrying homozygous recessive allele, which is susceptible to bacterial plant. And there are two alleles, small a and small b, same marker loci, flanking this target locus in recurrent parent. So capital A and small a are two alleles of this marker locus. Capital B and small b are two alleles at this marker locus. They have to be polymorphic. And that can be identified by any other population or the F2 population, any other population. The distance between this target locus and this markers have already been identified. That's a prerequisite for marker-assisted backcrossing if you want to include recombinant selection in your program. Now we'll develop F1 from these two parents. So it will have one homozygous chromosome from this, another homologous chromosome from recurrent parent. Here, of course, here this dominant target locus is present along with capital and capital B allele, and here this recessive allele is present from recurrent parent with small a and small b on either side. Now we backcross this F1 with the recurrent parent. And we evaluate the backcross 1 progeny. Here we perform foreground selection. We utilize tightly linked marker for selection of this target locus. And we also perform recombinant selection. We utilize these markers for identifying the recombination. So out of so many cases, in few plants, you may see that this target locus is present, but this region is now present with, this region is present from recurrent parent. Small b is present instead of capital B allele of this marker locus. So that indicates that recombination has occurred during F1 meiosis in this region. So that now this portion of the chromosome due to recombination is from recurrent parent. And in this generation, we reject all the plants which are showing this kind of constitution, homozygous for small a and small b, and the target locus. This is completely similar to recurrent parent. We don't want this. We also reject these kind of the progenies, which are having the normal chromosome from recurrent parent plus normal chromosome from donor parent. That means the chromosomal constitution without recombination. We are interested in this. Then we select these progenies. There might be only few plants, 1 to 5 plants. We select those plants, and then we backcross this plant with recurrent parent again. So that then we'll get backcross 2F1, backcross 2 progenies. In backcross 2 progenies, in backcross 1 we have focused on this recombination. In backcross 2 we'll focus on recombination on other side. So now you can see we'll reject the plants which are having the similar constitution to recurrent parent. Why we'll have backcross 2 progenies with a similar constitution? The gamete from backcross 1 progeny may carry this chromosome without any kind of segment from donor. And the gamete from recurrent parent will always, because it is homozygous, it will give this segment. We reject this. We also reject this kind of the constitution because we have already applied selection for this in backcross 1. We don't select this. We are only looking for those few individuals where, apart from recombination on this particular side, recombination has occurred this side. Now, where this recombination has occurred? This recombination occurred in backcross 1 meiosis. The meiosis of backcross 1 progeny, so that now we have the progeny which is having almost all the regions from, for this particular homologous pair, where our target locus is present, all the regions are from recurrent parent, plus the target locus is from donor parent. And we need to remember that still there is a heterozygosity for this target locus. But now for this marker locus, this marker loci, 2 flanking loci, they are homozygous for small a, small b, exactly similar to the recurrent parent plus our target. So here also in backcross 2, we apply foreground selection and recombinant selection. And if required, we can also apply background selection here, where we focus on other marker loci, except this target locus. We focus on other marker loci for deciding the recovery from recurrent parent. Now again we backcross this backcross 2 progeny, selected backcross 2 progeny, this one only with recurrent parent. So ultimately we'll be selecting the same individuals, small a, small b. Here also we apply foreground selection and we apply background selection to make it, to identify the individuals with maximum recovery from recurrent parent. And then we carry out selfing. You might have selected, let us say, 2 to 5 individuals out of 250 or 1000 plants. So here you can see we are applying selection for target locus, that is X, and definitely these are going to remain same because now the constitution is not going to change. When you utilize this backcross 2 progeny for crossing with the recurrent parent, there is no chance that capital A and capital B may be present here because you are not utilizing that in crossing program. So you will not find that in next generation. So here we apply selection for target locus, that is foreground, and we apply selection for background loci. Here 128 background loci have been utilized. This is rice. So there will be 12 chromosomes total to be focused on. And when you produce backcross 2F2, now this heterozygous locus may be in homozygous condition. So we apply selection utilizing codominant markers. We apply selection where both these recessive alleles are from recurrent parent plus the target locus from donor parent in homozygous condition. So you see in this backcross 3, we also reject this constitution, which we have selected in backcross 3. We reject this because in backcross 3 F2, where we have carried out selfing of backcross 3 progenies, we only want homozygous condition. We want homozygous condition for this target locus. So we reject heterozygous individuals. And then this is our final outcome, where the target locus is present. We have minimized the linkage drag, and by utilizing background selection, maximum loci or 99.9% recovery of the recurrent parent can be achieved. Now the third component, background selection, which I also partially explained in previous slides. So for background selection, we utilize the markers which are unlinked to the target locus, because in background selection, we are focusing on the recovery of recurrent parent, not the target locus. Target locus is from donor parent, and the background genome must be from recurrent parent. So we are applying selection for the microsatellite markers generally. SMP markers can also be utilized. So these polymorphisms are utilized to find out the recovery from recurrent parent. So how we can calculate the recovery? Maximum number of the allele should be from recurrent parent. Arrows are indicating the positions of these markers on the chromosome. And by general agreement, minimum 5 markers should be chosen from each chromosome. So if we talk about rice, there are 11 chromosomes, or 12 chromosomes, sorry, 12 in haploid constitution. So 5 markers per chromosome, it means 12 multiplied by 5. We need at least 60 markers for background selection. We can increase that by 10 per chromosome if cost and time permits. Funding opportunities are available, then we can make it 120. And these 5 or 10 markers you are choosing per chromosome must be equally spaced on the chromosome. 5 doesn't mean that you only focus on shorter arm. Otherwise, longer arm may be from donor parent, and that will decrease the recovery from recurrent parent. So we apply background selection utilizing the polymorphisms or markers which are not linked to the target locus but unlinked to the target locus. So we can accelerate the recovery of recurrent parent. So this is the conventional way. We need 6 backcrosses to achieve 99.2% recovery from recurrent parent. But with marker-assisted selection, it is possible within backcross 3. How it is possible? So marker-assisted selection or markers are not responsible for this recovery. By markers, we only apply selection. And we know that selection does not create variability. So 87.5% recovery, 93.8% recovery in backcross 3 itself. This is because of the recombination between the chromosomes of P1 and P2 in F1 meiosis BAC X1 progeny meiosis or BAC X2 progeny meiosis, the recombination is going. So theoretically, we know that in F1, there will be 50% contribution from donor and recurrent parent. BAC X1, there will be 75% contribution from recurrent parent. But each individual does not have 75% contribution. The contribution may vary from 75% because of the recombination between the chromosomes of P1 and P2 during F1 meiosis. So there might be some plant in BAC X1 progeny itself which are having more than 75% contribution or more than 97% contribution from the recurrent parent in BAC X1 progeny itself. So utilizing these markers, foreground, background, recombinant selection, these components help to identify the individual carrying the target locus with minimized linkage rate and having the maximum alleles from recurrent parent. And that's how within BAC X3 we can select the plant which is having 99.9% recovery from recurrent parent. Within BAC X2 also it is possible, plus our target locus with minimized linkage rate. So this is what I was talking about. In conventional BAC X's, we are selecting the individuals on the basis of phenotype. So we may be selecting many progenies from BAC X1 or BAC X2 for BAC crossing program. Now these plants have different contribution from recurrent parent. Some may have 75, some may have 80, some may have 82. So that's why it takes longer time to recover genome of recurrent parent. But if we apply marker-assisted selection here in this BAC cross program, that is marker-assisted BAC crossing, then we will select a progeny which is having the target locus from donor parent and maximum alleles from recurrent parent. So that's how the time required to recover the maximum contribution from recurrent parent is reduced, and it can be done within BAC X2 or BAC X3. So now just imagine if you have this SSR markers, marker linked to target locus and flanking markers. If you have this information, then you can achieve this within 5 seasons. Parents, you cross. In the next season, you generate F1. In the third season, you generate BAC X1 progeny. Fourth, BAC X2 progeny. And 5, BAC X3 progenies. So 5 seasons are required. Now the rice, if you talk about, there are certain crops you can take them twice per year. So within 3 years, you will be coming with the variety. So a PhD student can develop a variety with marker-assisted BAC crossing program. And if you incorporate speed breeding in marker-assisted BAC crossing, then in rice, 7 to 9 generations can be taken within the year. And here our selection is not based on phenotype. Selection is based on genotype. So if you apply speed breeding within 1 year, you can develop the variety, combining speed breeding and marker-assisted BAC crossing. So this is the example for, just you go through the slides for transfer of the flood tolerance, which is done by sub1 gene. It was transferred from this donor parent to Swarna, which is a popular mega variety. And it was also transferred to Samba Masuri also. So there is a phenotypic screening for submerged tolerance, and the BAC cross progenies are BAC cross to the recurrent parent, and simultaneously genotyping is also performed. And then once the desirable plant is selected in BAC X2 F2 population, which can be developed by selfing the BAC X2 individuals, selected individuals on the basis of genotype and phenotype, they are increasing the seed quantity of these plants. So this is the scheme followed for transferring sub1 gene from this particular donor to Swarna. So this is the F1 produced by crossing Swarna with this donor. This F1 was BAC cross to Swarna, which is recurrent parent. Out of 697 BAC cross progenies, they found 21 recombinants. So they are applying recombinant selection here. 376 had sub1 gene. So 376 had target locus. This information was obtained by foreground selection. And out of this 376, 21 were recombinant on one side of the target locus. So here we are talking about recombinant selection. So out of this 21, they selected a parent having the maximum similarity with the recurrent parent and plant number 242 was backcross to Swarna. And then there is 320 backcross to progenies. Out of that, 158 had target locus, foreground selection is performed, and 5 were recombinant on another side of the target locus. So out of these 5, one plant was selected, 227, which is showing maximum recovery from recurrent parent, and it was backcross to Swarna to produce backcross 3 progenies. They selected 18 plants having sub1 gene and maximum recovery from recurrent parent, and flanking alleles were from Swarna. And then they served this plant to achieve that in homozygous condition. And that's how sub1 gene was transferred to Swarna variety by Marcus and backcross program. This is just the scheme. Now you will be able to understand the scheme. Mostly in backcross 1, we perform foreground selection and recombinant selection. In backcross 2, both. We can also perform background selection. In backcross 3, we perform foreground and background selection. So this is the locus governing submerged tolerance, sub1 locus on 9th chromosome rise, and other genes are from recurrent parent Swarna. So that's all for today. Tomorrow I will consider markers gene pyramiding, markers recurrent selection and genomic selection, and other sequencing methods based on NGS. So that's all what I wanted to say today. If you have any question, then we can discuss that. Yes, flanking marker information we can obtain by gene mapping. As I told you earlier, we can develop mapping populations from these 2 parents, or this marker information may be obtained from any other sources like online databases, or you have to find out the polymorphism between these parents, and utilizing any mapping population, you have to identify markers which are flanking the target locus. So by gene mapping only, we can have that information. Or if some information is available related to the target locus and the flanking markers, then that information can be utilized from any other variety. And if that holds true for your recurrent and donor parent, then you can utilize those flanking markers in your backcross program. There are many ways we can do that. Mostly when map-based cloning is performed, which is also known as positional cloning, generally locus under consideration, which is much more important, like sub1, generally high resolution maps are also available for that locus. So you have to review those papers where high resolution maps have been prepared. High resolution maps means we have identified many markers closely situated to the gene of interest, to map-based cloning or any other method. So you have to review those things, and from that you can get the information. And you have to find out the flanking markers to the locus which are polymorphic in your parents, which you want to utilize as recurrent and donor. That can be utilized. If that information is not available, you have to find out on your own by utilizing the mapping population or F2 population. You have to find out the flanking markers to the target locus, and then you can utilize. That is a prerequisite for recombinant selection. Disease screening is utilized to be sure that the target locus is which is present and identified on the basis of molecular markers it is actually responsible. I mean, it is all the same plant is also phenotypically giving the same response as expected because the marker is present. Just to have assurance that the Bacchus progeny selected is actually phenotypically resistant to the disease, or phenotypically it is expressing desired trait. To be sure, phenotypic screening is applied. Phenotypic screening, it depends whether you apply it or not upon breeder's will, breeder's desire. If someone wants to be extra sure that once marker has identified particular target locus, it is phenotypic. It will be definitely phenotypically expressed before backcrossing program phenotyping is done, just to have extra surety and confirmation. Once you prepare marker maps, or again, as the question was asked, that how we identify the flanking marker, how we get the information. Similarly, you can search for the papers where this marker information is available in your crop. Suppose if it is rice, then there are many papers available where saturated marker maps have been developed. So chromosome wise, there are markers identified, and the distances in centimorgans have been identified. So you can have a scale, your our normal scale, and you can identify the papers where these marker maps have been prepared. By utilizing the scale only, you can find out the equally spaced markers, and you can utilize those markers. When you do that, you have to, before going to the backcrossing program, you have to make sure that those markers you have identified equally spaced on the chromosome, those markers are polymorphic in your parents. Then and then you will be able to utilize that marker. Simply you have to put the scale over the marker map of every chromosome, and you can identify equally spaced markers. Because when marker maps are prepared, if two polymorphisms are closely situated, then the distance between them would be less, and they will be placed together or closely each other on a marker map also in that paper. And if two polymorphisms or markers are situated apart, then they will be placed away from each other. So simple scale you can utilize to identify equally spaced markers. A good question. And just to have that idea, you can search in Google molecular marker maps in rice. You will get many paper, or saturated linkage maps in rice. You will get many paper. If you look into that paper, there will be 12 linkage groups, and each linkage group you will be able to see. You can identify equally spaced markers. Yeah, welcome. If there is any question, we can deal with that. Okay, then we'll continue tomorrow from 11 to 12. Thank you very much.